Hi, my name is Andrea Pacheco. I'm from Peru, and my work is titled In Silico Characterization of Delta Conglutins in Lupinus mutabilis. Lupinus mutabilis, also known as tarwe, is a crop of major importance in the familiar agriculture in the Andean region. Lupins are gaining global interest due to their nutritional value and has been also associated with playing positive roles in health in areas such as combating obesity, diabetes, and cardiovascular diseases. The main storage proteins in lupins are known as conglutins and have been classified into four groups: alpha, beta, gamma, and delta conglutins. Here we focus on delta conglutins. These proteins belong to the 2S albumins family, and 2S albumins from other plants have shown antifungal, insecticidal, and antibacterial properties, receiving considerable attention due to their potential use to control agricultural diseases and for the production of new antibiotics for the treatment of various human infections. This study aimed to identify and characterize delta conglotin transcripts and protein sequences from Andean lupin seeds transcript. First, we downloaded raw reads from the ENA database. These reads were preprocessed and assembled using FastQC, TrimGalore, and Trinity. The redundant sequences were removed using CD-HIT. The final assembled transcriptome's quality was determined by using Boti2 and Busco. Delta conglotin sequences from different plant species were used to build custom databases. Once we had all these reference sequences, we identified delta conglotin transcripts in the Lupinus mutabilis transcriptome using BLAST. Then we generate some HMM profiles using the reference sequences of delta congrutin using HMMR program. Then we compare these profiles with Lupinus mutabilis open reading frames, which were obtained using our finder. The characterization of data conglutins, proteins, and transcripts were done using SMART, CD search, and PFAM to identify conserved domains, and MIM to identify conserved motifs. Geoterms were assigned to open reading frames of interest using BLAST2Go. For the phylogenetic analysis, protein sequences alignments were done using MAP. Then we used model tests IQ3 and ITOL to construct the phylogenetic tree. Lastly, 3D structure models were predicted using Rivera Fire 2, and null probability and multifold were used to choose the best predicted model, and PyMol to visualize the resulting protein structure. We identified six non-redundant delta congruent transcripts in the C transcriptome of Lupinus mutabilis. In Table 1, we can observe some characteristics of the identified sequences, physical and chemical parameters, including number of amino acids and geoterms. In Figure 1, we can observe a phylogenetic tree of the delta conglutine protein sequences from various Lupinus species, including Lupinus mutabilis. We can observe the conglutin 5 and 6 from Lupinus mutabilis grouped separately and also show some differences in their conserved motifs. Lupinus delta conglutins share a conserved domain, which is a trypsin alpha amylase inhibitor. Alpha amylases enzymes are essential to degrade starch during the seed germination, so the delta conglutine ability to inhibit it indicates a role in seed germination. Enzyme inhibitors have also shown an important function in pathogens defense. In Figure 2, we can observe a phylogenetic analysis of 2S albumins from 11 different plant species, including all the sequences obtained from the Lupinus genus and the recently identified delta conglutins from Lupinus mutabilis. In Figure 3, we have the best 3D structure model predicted. This 3D structure was produced by Rovera, and we used the Lupinus mutabilis delta conglutin 1 protein sequence for the protein structure prediction. The best model was selected using mold probability and mold fold. We can observe the predominance of alpha helices in the secondary structure. 2S albumins share 8 conserved cysteine residues. In this study, we observed that 2S albumins from Lupinus mutabilis have a high content of cysteines with an additional cysteine residue in all identified sequences. Cysteine residues involved in disulfide bonds are shown in violet. The 4-disulfide bond pattern has also been described in the majority of 2S albumins, and here we show in yellow the 4-disulfide bonds indicated by PyMol. In conclusion, we identified six non-redundant delta transcripts in the seed transcriptome of Lupinus mutabilis. We also determined that the lupin delta conglutin shared a common trypsin alpha amylase inhibitor domain, which has been involved in antibacterial and insecticidal activity in 2S albumins from other plants. The 3D structure of delta conglutin from Lupinus mutabilis predicted in this study shows the conserved pattern of 8 cysteines and 4 disulfide bonds, characteristic of the 2S albumin family. These results will be useful in elucidating the function and further biotechnological application of delta conglutin in Andean lupin. Thank you.